Microbodies!
Hey everyone, we are the Study Buddies. Welcome back to our channel. So, in today's video or biology edition, we are going to learn about the continuation from the previous video, which is Eukaryotic Cell Part 2. If you haven't watched the previous video, do watch it first and come back here for better understanding. In our previous video, Eukaryotic Cell Part 1, we had discussed about some key organelles such as nucleus being the control center of the cell, ribosomes which are the site for protein synthesis, and the entire ER or endoplasmic reticulum which consists of rough endoplasmic reticulum or RER for protein transport, smooth endoplasmic reticulum or SER for lipid production and detoxification of drugs, Golgi apparatus or GA for modification, sorting and packaging of proteins and lipids, and lastly, lysosomes functioning to destroy unwanted particles with hydrolytic enzymes in it. In continuation with that, the next organelle in eukaryotic cells that we are going to look at are microbodies. Microbodies are basically vesicles that contain specialized enzymes that perform specific metabolic functions. So, there are several types of these microbodies in eukaryotic cells. Firstly, we have peroxisomes. Peroxisomes are found in both plants and animal cells. They were one of the first organelles to be discovered and characterized in eukaryotic cells. They have a simple structure consisting of two major parts, an outer layer of single lipid bilayer membrane and a crystalline core. The image to the left shows the basic structure of the peroxisome with a lipid bilayer surrounded by a plasma membrane and a condensed crystalline core in the center. The core contains a variety of enzymes for catalysis of cell functions, mainly metabolism. Typically, the most concentrated of these enzymes, catalase or urate oxidase, can become so high in number that they aggregate to form crystalline core in the center of the organelle. They contain enzymes used to break down fatty acids. Unlike lysosomes, which we learned earlier, containing hydrolytic enzymes, enzymes in peroxisomes are actually synthesized by free ribosomes and transported into peroxisome. Its main function is to oxidize fatty acids or generally lipids into water and oxygen. These are usually found in liver cells where some produce bile salts from cholesterol and others break down fats. As in plant cells, peroxisomes oxidize fatty acids into sugars needed by growing plants. Next, we have the vacuoles. Some may get confused with vesicles and vacuoles, but they are not the same. Vacuoles are like sacs and are quite larger than vesicles, and they tend to be in one place only, whereas vesicles tend to travel to different places. A vacuole is an organelle in cells which functions to hold various solutions or materials. This includes solutions that have been created and are being stored or excreted, and those that have been phagocytized or engulfed by the cell. A vacuole is simply a chamber surrounded by a semi-permeable membrane which keeps the cytosol from being exposed to the contents inside. Because vacuoles are surrounded by semi-permeable membranes, they only let certain molecules through. In some organisms, they would have contractile vacuoles for getting rid of excess water from its body, such as in Paramecium. Being in a watery environment, water would constantly enter Paramecium's body. So, that is when contractile vacuoles squeeze themselves, expelling the water out of them. As you can see in the picture, water enters the contractile vacuoles, and once it reaches a definite size, vacuoles are able to squeeze themselves, hence expelling all those excess water in the organism as well. As in plants, a vacuole's function is to store water, sugars, salts, pigments and toxics. Plant cells have a central vacuole filled with a watery fluid called cell sap. It maintains the cell's turgidity by maintaining hydrostatic and turgor pressure. lack of water in the vacuole, the plant will start to wilt because it has lost the pressure needed to hold it turgid. Looking at this picture, we can clearly see that the cell that has a big, expanded vacuole is turgid and fresh because the cell sap in the vacuole exerts a certain amount of pressure on the cytoplasm of the entire cell, pushing all of its cytoplasm throughout the cell. While the cell that has a shrunken and small vacuole looks wilted and unhealthy with the lack of water in the vacuole. This explains why plants look wilted when water is not supplied to them. Next, life is possible only because a constant input of energy maintains the structure of the cells. In that case, chloroplast and mitochondria are two eukaryotic organelles specializing in converting energy to a form that can be used by a cell. Animal cells contain only mitochondria, while plant cells have both mitochondria and chloroplast in them. The energy generated by these two organelles are in the form of ATP molecules. The energy of ATP is used for various other biochemical processes as well. First off, we have the chloroplast. It is surrounded by a double membrane which includes an outer and inner membrane. Stroma is the semi-fluid region enclosed in the double membrane. Thylakoids are disc-like sacs containing enzymes and pigments in them. A stack of thylakoids is known as a granum. These grana are connected to each other, forming a thylakoid space within them. Chlorophyll is a pigment that provides the significant green color in most plants, along with several other pigments that can be found in the thylakoid space as well. Chlorophyll is responsible in capturing solar energy and converting it into chemical energy or sugar for cells usage. This process is referred to as photosynthesis. As a matter of fact, chloroplast is a plastid, meaning it is a plant organelle surrounded by a double membrane and have varied functions. Some other examples of these plastids are called chromoplasts, containing color pigments for red, green and orange colors, and leucoplasts, synthesizes and stores starches and oils. Moving on to mitochondria, they are definitely much smaller than chloroplasts. Generally, mitochondria is known as the powerhouse of a cell due to its ability to generate energy in cells. A mitochondria has a double membrane system too, with an inner membrane folded into structures called cristae and projecting into a matrix. A matrix is a semi-fluid environment containing mitochondrial DNA and ribosome. The cristae is folded in order to maximize the surface area for reaction to occur. The enzymes in the mitochondrial matrix break down carbohydrates and other nutrients to release energy in the form of ATP molecules. This process is referred to as cellular respiration. If you look at this picture, it explains how chloroplast and mitochondria work together in generating energy, especially in plants. During the day, chloroplast does its job by using water and carbon dioxide in the presence of solar energy and chlorophyll to release oxygen and glucose. Mostly towards the night, when there is no sunlight, mitochondria does most of the job by using up oxygen and glucose, releasing carbon dioxide, water and energy. Animal cells, on the other hand, being independent of a light source, does not require a secondary source of energy generator like in plant cells. The next thing we are going to discuss is called the cytoskeleton. Cyto basically means cell and skeleton is like a framework that supports something. Just like the bone framework in our body, cells also need a system and organelles that maintains its shape, movement and provides structural support. Oftentimes in a picture of a cell, we won't get to see the cytoskeletons as they are super tiny indeed. Generally, cytoskeletons contains actin filaments, intermediate filaments, and microtubules. First in line, we have the actin filaments. These are long and extremely thin, flexible fiber networks and have bead-like structures as well. It lies under the plasma membrane, providing structural support. An exemplary situation where actin filaments play their role is when an amoeba moves over a surface with its pseudopods. The alternate contraction and movement happens when the actin filaments work together with a motor molecule called myosin. With the expenditure of ATP, myosin molecule pulls the actin filaments along its way. We will learn more about this process of actin filaments and myosin working together on our future videos on muscle contraction. Nextly, based on its name, intermediate filaments are intermediate in size between actin filaments and microtubules. They assemble a rope-like structure. As seen in the slide, intermediate filaments contract and relax as shown to bring about movement. Some of the main functions of it are to support the nuclear envelope and form cell-to-cell junctions to hold cells together. If not for them, our skin would just peel off when get pinched. Lastly, let's look at microtubules. It looks like a hollow cylinder made up of two globular proteins, namely alpha-tubulin and beta-tubulin. Typically, they interact with motor molecules called kinesin and dynein to move the molecules as you can see in the picture. The two vesicles are the substances that are being transported by the microtubule. With the expenditure of ATP molecule, kinesin and dynein acts alternately, bringing about movement in the substances. Microtubules also play their role in cell division as well. If you may recall about mitosis and meiosis, we have a structure called centriole that is involved in this process. Microtubules in the centriole disassemble into spindle fibers or these string-like structures during cell division to distribute chromosomes in an orderly manner. After the process, they would just reassemble again into their former arrangement. And with that being said, this will be the end of the video about part 2 of eukaryotic cell. Thank you everyone for watching this video, and I really hope you've learned something new today. If you like this content and want more of it, do like this video, share it with your friends and family, and subscribe to our channel. Don't forget to hit the bell icon as well. If you have any doubts regarding any subjects, do comment your question in the comment section and we will help you to answer it. Thank you everyone and take care. Goodbye.